Hi, my name is Alex Fournier-Level. I'm a senior lecturer at the University of Melbourne in the School of Biosciences. And today I'm going to talk to you about the adaptive evolution of annual ryegrass in Southeast Australia. Annual ryegrass is currently the worst weed in Australia owing to its incredible capacity to develop herbicide resistance. It is currently the species that has developed single resistant to the most modes of action. On top of my head, 19 modes of action are being faced harmlessly by ryegrass, which is pretty astonishing. In recent years, we've developed novel genomics resources for this species, which is a diploid species, mostly outcrossing, henceforth bearing a lot of diversity with a large genome, 7 chromosomes, 2.4 gigabase. We've generated a chromosomal level assembly. It's a pretty classic standard grass genome, essentially made out of repeat elements, among which 30% are transposable elements, and a rather large number of genes for a diploid species with 57,000 genes. It is essentially native to the Mediterranean basin, although its native range spreads a little bit beyond from the Atlantic face up until the central Middle East and a little bit beyond. And interestingly, it is sharing this space with a sister species that is the perennial ryegrass. And this perennial ryegrass occupies the northern end of the distribution range of rigidum. So they largely overlap across northern Spain, France, and Germany, where the solid triangles are, whereas rigid ryegrass thrives more essentially in drier areas, owing to its capacity to produce seed and withstand warm summer as seed. And the seeds actually require some exposure to elevated temperature before they can germinate the next year. So it becomes a particularly noxious weed in winter cropping. And in the context of Australia, most of grain cropping happens over winter with absolutely no way of growing a summer crop, given the level of drought and high temperature. Rigid ryegrass has is becoming a cosmopolitan weed, having invaded numerous areas across the globe, across all continents. So it is a slightly too significant problem in California, as well as in Latin America and South Africa. But the place where it has become really a constraint to yield and productivity is Australia, where it pretty much covers the full extent of the arable zone. So in terms of tackling the issue, we've committed to genomics research since 2017 in my lab, and we've generated a good reference genome for that species. This was made using long-read nanopore sequencing technology on a Promethean platform, together with some Illumina whole genome sequencing, to get to the point where our total genome assembly is really close to the estimated genome size based on cytometry. We are getting to an L90 of 7 chromosome, L54, which is what expected. Strikingly, we do observe very little gaps. So we've got a good reference genome, and transcript support gene models reach a tally of 57,000, which is rather large. We set up in place a compare genome workflow to conduct comparative genomics analysis. And upon analyzing the differentiation between the reference genome for Lolium perenne and or reference genome of Lolium rigidum, what we find strikingly is that those species are indeed rather close. However, we did observe a striking expansion of genes in the Lolium Lolium rigidum background with over nearly 4,000 extra gene in Lolium rigidum compared to Lolium perenne. And those correspond essentially not to novel genes, but with multiple orthologs, orthogroups. So basically, these number of expanded genes rely on pre-existing copied genes. So it's definitely not a loss in Lolium perenne that has driven this expansion in Lolium rigidum, although in the context of Lolium perenne, we did see rather heavy contraction of gene families. So that's interesting to note that both species have really diverged in terms of gene retention. So for today, what I'm going to present is mostly aim to deliver a molecular diagnostic of herbicide resistance to farmers, farming groups, and agronomy advisor. So we're not going to really focus, or we're really not going to focus on talking about gene function and characterizing the genetic basis of herbicide resistance from a functional perspective. Here, what we're trying to achieve is to use resistance variation present in the field, match it against genotype data that we're generating in the lab, train quantitative genetics model with some level of cross-validation to actually get to the point where farmers or advisor can ship us a sample from the paddock for which we conduct DNA sequencing in our lab and produce a herbicide resistance prediction report that could be used to better manage the weed infestation in the paddock. So to do that, we had to deploy an integrative framework that is rather broad, including characterizing herbicide resistance diversity through survey collection, phenomics conducted in the greenhouse, and I'm going to present you the outcome of this trait level analysis. We also had to build a strong data set in terms of genotyping of this population, and we are going to present the tools we're deploying to build this genomics data set. And finally, I'm going to cover the genetic mapping techniques we're deploying, either through GWAS or up to genomic prediction frameworks, which is what is commonly used by breeders to gain insight into trait variation, just relying on genetic variations. So in this context, we're going to start with discussing how we surveyed population and tested herbicide resistance in the lab. So we've started our efforts early in 2018 with a broad collection of 60 population, collected another 60 in 22, and got the supply of many population from Peter Boutsalis at the University of Adelaide. And now we have, you can see the different survey efforts in the different colors, but now we're covering quite a broad regional scale across Southeast Australia. So I'm here centered in Melbourne. That's basically my back garden, and we're trying to cover as broad of a region as we can in the grain cropping belt of Australia. When we surveyed plants, we've been focusing on harvesting seeds for a total of 259 population sample between 2018 and 22. Generally, when we get samples in our lab, we process the sample to dehusk the seeds. We treat them to avoid excessive infestation in bacteria and fungi. We then conduct germination assays in plates in growth cabinets before transferring 42 individuals per tray, replicating 2 trays per population. So our workflow yields to analyzing 84 individual plants in each population, and for 84 individual plants. We've been harvesting the leaves, grounding them for sequencing, and then setting up these 84 individuals per population multiple times to spray with 10 herbicides and get a database of herbicide response across all population for these 10 herbicides. So with these 10 herbicides, it was really relevant to us to test a level of multi-resistance and correlation in herbicide response. And what we've been able to identify is a set of relatively cohesive clusters of resistance, and typically the most striking of them is a strong correlation between resistance to glyphosate and resistance to paraquat, which are two herbicides that are used in tandem in multiple management practices. And what it highlights here clearly is that we observe correlation between herbicides that are co-used in specific management practices rather than picking up on the same or similar mode of action across herbicides. Although I need to disclose that I'm not a biochemist and I really, I'm only an enthusiast here. You guys probably have a better background on the specific topic. So based on this correlation, what was interesting to us is to map a source of resistance across the landscape, and that's what we've been doing either for multi-resistance or for individual herbicides. So here I'm presenting the different herbicides for which we identified non-random distribution of herbicide-resistant sources over the landscape, and you can see that glyphosate has a main hub of resistance together with a couple of hotspots, and you can see that the main source of resistance also applies to peroxysulfone or paraquat. Atrazine having a different pattern typically of resistance, more centered around a southern region. And atrazine is still widely used in Australia. So what we could do with these data and this landscape explicit mapping of resistance is to build environmental niche modeling for the resistant populations. And what was interesting in this analysis is, as you can see in blue here, we've got among all the predictors, the environmental predictors, which combine soil characteristic retrieved by SARO and modeled at very high resolution. So we've got like a 90-meter resolution grid over the entirety of Australia. So that's really a great asset for soil characterization across multiple depth, as well as the most commonly used bioclim data from WorldClim. And what this analysis emphasizes is that the predictors of resistance, and particularly to glyphosate and atrazine, clethodim to a lesser extent, is that soil characteristic that really drive the herbicide treatment efficacy also drive the presence of resistance. So you've got the most important variable in predicting the emergence of resistance is organic carbon in the soil, together with water content at a relatively shallow surface, as well as precipitation in the wettest quarter, precipitation in the coldest quarter, which are in fact variables that predict high level of organic matter and wet soils, which we know lead to inefficient herbicide treatments, which at low dose is expected to push levels of resistance in the paddock. So this analysis made sense, and this is a good asset to be coupled to our genomic analysis of these populations. However, this is done on a proportion of surviving plants among the 84 individuals planted per population, which is a rather coarse and not very informative from a plant physiology perspective. And what we've been developing is a colorimetric index of resistance to provide a quantitative estimate of resistance at the single plant level. So to do that, we photograph all plants after spraying them, and we process them through a computer vision workflow we called Instagraminoid. This has been published in 2019. And clearly what we can see is that indeed a dead plant being brown, a resistant plant being green, we can distinguish quite powerfully resistant from susceptible using that technique. So we generally have 95% of correct classification with a single set of picture after 21 days. But what was interesting to note is on top of just classifying correctly susceptible versus resistant, we were able to note that among the ones that don't survive, we've got like a bit of a bimodal distribution from the very susceptible plants to the nearly or getting ready, so to speak, to become resistant. So we really believe that this colorimetric index provides a quantitative estimate of resistance, and classifying this population as susceptible is a good thing, but being able to measure how close they are to hitting the resistant threshold is interesting, and that's what this metric can bring. And typically, when you analyze individual plants versus proportion of resistance in a population, in the context of the panel A here, resistance proportion was not able to identify very powerfully cross-resistance between glyphosate and 3 other herbicides we've tested, whereas using this colorimetric index based on our image analysis, we are able to pick up subtle quantitative correlations that could inform management better, because if you've got an index that is close to resistance, you can pick up the emergence of resistance maybe before it's too late and the resistant population is established in the paddock. So we're pushing that logic, and we are now equipped with a 3D multispectral scanner that is going to be able to measure multiple traits beyond the visible spectrum. So this is a work in progress. But this is the type of capability we now have acquired at a relatively high throughput in the coming couple of months. So with this data set, now we're going to focus on the genotyping of population of ryegrass to conduct genomic analysis. So I've got 2 main things to advocate for. First, the use of pool sequencing when it comes to characterizing weed populations, because we've been able to demonstrate quite clearly that in the context of, I'm going to break down this graph, in the context of sampling most of the landscape, so those correspond to the red dots here, the top panel represents the true positive rate in terms of how easy it is to pick up a herbicide-resistant gene versus not. And FPR, false positive rate, which means you are identifying genes that are actually not involved in herbicide resistance. We could see that when we sample quite extensively the landscape, basically individual sequencing, so every individual of the population versus pool sequencing cost 10 times more. So the effort committed to individual plant sequencing needs to be 10 times that invested in pool sequencing to get to the same point. And in the context of the blue dots here, which means what we generally do actually, we sample only a fraction of the landscape. Here we're modeling somewhere between 10 to the 20% of the populations are being characterized, and in that case the improvement of pool versus Indy is only 3-fold with a maximum ratio of 0.33. However, that still represents a substantial gain, and really there's, I believe nowadays, very limited reason in the context of developing genomic prediction model and population genomic characterization. There's very limited reason why I should deploy INDI-seq versus pool-seq, because at the population level, my predictor becomes continuous allele frequencies that are actually the currency of most population genomics analysis. Given the size of the ryegrass genome, that is not enormous if you compare it to hexaploid wheat, but still large enough not to allow us to deploy whole genome sequencing across all of population, we've deployed a reduced representation approach, such as that presented by Kaio in his talk last month. We've optimized the pair of restriction enzyme we used, and we identified that based on the mapping on Lolium pyrenea, because at that time we didn't have the reference genome for Lolium rigidum, we identified that M-cell and NSI1 correspond to the best couple of enzymes. So you've got a frequent cutter and a rare cutter that leads to the best coverage in terms of depth of our genome. So that's what we adopted. So we're coupling ddRad with pool-seq to generate allele frequency data genome-wide efficiently in terms of cost, because individual samples are not very precious, but population level information is critical from a farmer's perspective. We validated that in this context, for the 119 first population with genotype, we were able to generate a database of 272,000 SNPs that are at a coverage greater than 10x across all population, that represent one SNP every 8.8 kb. And we really covering quite evenly all 7 chromosomes with relatively limited variance in terms of depth. So we've got pretty much of an even coverage, both in terms of breadth and in terms of depth, which is satisfying, and this is what I've been presenting in September at the Plant and Animal Genome Conference under the International Wheat Genomics Consortium symposium run by Todd. So at that conference, what I had presented is a preliminary analysis showing that indeed, when we're mapping pairwise genetic distance and pairwise geographic distance, we do observe significant but not really strong correlation, meaning that ryegrass populations are indeed isolated by distance, as expected, but not too much, with indeed relatively high diversity and really low patterns of differentiation, because the nominal value for FSTs are quite subtle. However, what was interesting is that when you overlay the distance, distance in resistance, so how different two populations are in terms of resistance to glyphosate only from 0 to 100%, we could clearly observe that actually FST, so population differentiation, was also quite similar to herbicide-resistant differentiation between susceptible and resistant, which was really interesting to us. Here are different ways of producing this graph, but clearly we observed that, sorry, I'm starting with the middle panel, with observing quite a strong correlation between glyphosate resistance and FST. And interestingly, but expectedly, we do observe that resistant to glyphosate is negatively correlated with heterozygosity, which could be a sign that resistant population are slightly less diverse, and that is evidence for glyphosate resistant being selected for in weedy populations. So that could fit the preconceived model we had so far. So we've reconfirmed all these analysis by first doubling up our population sizes and increasing the sequencing output, running a couple of Novasic flow cell. So now we have increased quite substantially the set of SNPs with genotype across population. So across the 234 population that we've been able to genotype, we generated over 50 million SNPs that are polymorphic with a minor allele frequency greater than 10% in at least 2 populations out of the 234, just to make sure that this is not a sequencing error, and set a cutoff to 10x across 50% of the populations. And we found a similar pattern as the one I had presented you before, but in this context, what we've noticed is some discrete clusters of differentiation. So the pairwise genetic distance between population was not as continuous as it was, but clearly we had here a set of population comparison that was very different from the rest of the cloud, and a second here cluster, and that was something that puzzled us. So what we conducted as analysis is trying to identify which populations were driving this differentiation, whether it was really based on the 2 pairs of involved in the comparison, or whether it was systematically the same population that generated the differentiation. And this later hypothesis was the correct one, because if you look for enrichment in a specific population or cluster, we noticed that the green cluster here is only relying on the comparison between the rest of our population and 2 samples that are most likely from a different species, and we expect this to be, in fact, mostly Lolium rigidum, which is quite hard to distinguish at the plantlet stage in the lab, and it's even in the paddock and as bigger plant may be better, but that I'm not going to blame it on the collection from the University of Adelaide, but indeed, as you can see, these misspecification were among the sample from the University of Adelaide. But what we could also tell is that there's a more subtle intermediate cloud of population comparison that were contributed also by specific population. And in fact, here our hypothesis is that these intermediate clouds correspond to hybrid populations, or when even more subtle, to minute introgression of Lolium perenne into a Lolium rigidum background. So when we remove these 2 clusters, the pattern of isolation by distance is reinforced, but we clearly are starting to see the importance of gene transfer, hybridization, introgression among the sister species that actually correspond to a species complex. And I think it's going to be interesting with the development of more resources in the 3rd species of ryegrass that is problematic, which is Italian ryegrass, and see how we could analyze this data even better. Conducting the same analysis and imposing distance between herbicide resistance among pairs of population on the graph, what we observe is that the cloud that is mostly constituted of comparison between Lolium rigidum and Lolium perenne doesn't show any particular trend in herbicide resistance, and in that case with respect to glyphosate. We noticed that among the 2 populations of Lolium perenne, one is resistant to glyphosate, the other one is not. So there's not much of a trend. But here, among the intermediate, where we suspect hybridization and introgression, we did observe a quite dramatic enrichment for herbicide resistance with respect to glyphosate again, and that really prompted us to go towards identifying patterns of introgression as potentially the basis for resistance, and I follow up on that in my opening conclusion slides. Now, taking this data point and spreading it at the genome-wide level, we are able to identify loci in the genome where herbicide resistance differences are particularly well explained by genetic factors, such as that one on the middle of chromosome 6, whereas in general, we observe that the level of geographic differentiation is kind of spread across all the genome. So specific zone where the differentiation due to herbicide resistance far exceeds what is expected under a null model should point us at interesting region for herbicide resistance. So now I'm going to move to the part that was really the main aim of the research program in my lab, which is generating genomic predictions of herbicide resistance for a range of herbicides in Southeast Australia. And what we've been able to achieve is a overall production accuracy around 60%. We initially only focused on genetic models, but we also apply prediction models only relying on soil and climate data in similarly as to what I've shown you in the first part of my talk. And we could show that in the context of glyphosate, adding soil data and climate data only marginally improves the genomic prediction models that are still around 60% regardless. So the best model relies on both, but genetics has more of a factor, which is a good thing for a genomic prediction model. However, one issue for practical implementation of genomic prediction model is that the best performing model, and we tried a wide range, is not the type of parsimonious penalized model that only uses a fraction of the genome and then can be deployed more steadily because you need less marker. In our case, the best genomic prediction model for weeds are GBLUP type of models, and these models require the use of all markers genome-wide, and this means that it's quite sensitive to missing data and error. You need to have a pretty steady marker coverage when you regenotype your new population and make sure that your frequency estimate is quite accurate for every single marker, which is a bit of a constraint. However, these models seem to apply across most herbicide for which we were able to generate predictions. And I'm not presenting them all, but I'm taking an over time perspective where you can see that prediction accuracy, it's between 60% and 66% from atrazine, which is a quite old herbicide, Intercept, which is a mix of imazamox and imazapyr, up till a more recent one, Overwatch, bixlozone, for which level of resistance, nominal level of resistance, is only 0.3% when we do observe over 40% resistance for some population to the Intercept, which is a commercial name of the formulation that we've used. So we could demonstrate that prediction accuracy is actually not necessarily requiring to have high levels of resistance in the population to be detected, but it's rather just about the standardized variance. So you need a good spread of resistance variation in the population, even at low level, but that just increases slightly the error bar for individual genotypes, but the overall prediction accuracy does not drop. So that was interesting because we were able to generate genomic prediction between populations. But one idea is that there's a lot of variability within populations. So we were interested in boosting our genomic prediction models using within population variation. And to do that, we've focused on a resistant population from Western Victoria and a susceptible population from Eastern Victoria, these two, forget about the third one, and used a technique that I had developed a bunch of years ago, which is called GWALFA, which is able to generate parametric estimates of pooled GWAS. So the subtlety of this method is that instead of just relying on sequencing the two tail ends of the distro, like most box segregant analysis do, what you need to do to deploy the GWALFA technique is to sample all the pools, so set thresholds for level of resistance and slice up the distribution of resistance within population. And that has the advantage of helping you to measure the allele frequency at the whole population level, whereas when you only focus on the two extremes, you're not being able to estimate the overall allele frequency. The underlying model is a maximum likelihood model that fits the underlying mixture of distribution between two alleles at a locus. It actually can be more than two alleles, but in most cases, we're using bimodal binomial SNPs. And the difference between the mean of the underlying distributions within the overall population is actually a measure of the allelic effect at this locus. So that's what we've been using in various frameworks to generate within population genomic predictions. And a proper scaled genomic prediction done in a traditional way yielded okay result across population. But what we've noticed is that a crude sum of all effects, alpha effects, multiplied by frequency, actually yielded very accurate predictions in terms of ranking. So you're not being able to get a percentage of resistance in your population like you would in the other model, but we could see that it doesn't work too well for terbutaline. Here are the red dots, but typically it's strikingly good at predicting glyphosate resistance within populations. And here you've got 10 dots corresponding to 2 times 5 pools for the two population. So this was pretty good and and we were pretty confident that our GWALFA GWAS was rather accurate. So that pushed us to analyze the pattern of genome-wide association and in our populations. And in that context, what we were able to see is that the GWAS peaks kind of matched, not all of them, but for some of them, dense in TAGIM-SD estimates. So TAGIM-SD measures overall patterns of selection based on site frequency spectrum with negative values corresponding to negative selection or purifying selection. And in that case, you see dense in TAGIM-SD that could be evidence for herbicide resistance loci to be selected for. So we worked a little bit more on this framework. This was something that we did last year. Also, interestingly, we did recapitulate within population the association we found based on our population genomic analysis of all populations, so that this mid-chromosome 6 locus definitely, it's quite broad, but definitely bears relevant variation that probably warrants investigation at the functional level. But what we've been doing is developing a modeling framework to test for convergent versus divergent evolution in herbicide resistance. The model is general purpose, but by contrasting fixation index FST and TGMSD and the width of TGMSD that decays with decreasing linkage disequilibrium, we were able to set null hypothesis for adaptation relying on de novo mutation, standing genetic variation, independent emergence, migration, or shared ancestry. And in our data, what stood out is that in most cases, the instance where these peaks deviate from the null hypothesis, it deviates from the null hypothesis of standing genetic variation. Oh, sorry. It deviates from the null hypothesis towards accepting the hypothesis of standing genetic variation being leveraged within population to promote resistance. So it seems to say that the pileup of resistance that already pre-existed in the paddock is the main driver of steady increase in resistance over time. So I think this is really an interesting point. I'm not saying that this is the only mode of action, mode of evolution, but our data at the current time supports this as the main mechanism of buildup of resistance in the field. So to wrap up here, we really not discovered, but rediscovered or confirmed that annual ryegrass populations are very diverse, not very structured over the landscape, with this complex genetic structure, which we believe can be partially driven by hybridization and integration with other species. This is something that we need to test formally. But we also noticed that genetic differentiation is indeed driven by herbicide exposure and a bit geographic distance. So all the components you would expect as driver of genetic structure are present in ryegrass. So from a bit of a simplistic model that herbicide provokes new mutations, so promotes this de novo emergence of mutation, and generally in major resistance gene, may not apply too much in the context of Australian ryegrass, but instead we're favoring a model under which standing variation is being recruited to build resistance over time across the landscape. So herbicide resistance is complex, and there's multiple mechanisms that are not exclusive. So it's just a matter, in fact, in my opinion, to integrate the different components of the model. So we still have the dichotomy between monogenic resistance of large effect that you can tag with a major marker and quite well characterized substitution at the amino acid of protein level. Those are hard to predict, their emergence being quite stochastic. So we still believe there's some of that, but what we're bringing to the table is the emphasis on polygenic resistance, where you've got subtle genetic changes that can be indeed captured by your genomic prediction model. So herbicide resistance over the landscape, to do the genetics of it, you need dynamic and multifactorial model, but population genomics is really relevant, and I think there's a big opportunity to do more of it across multiple systems to really gain insight and support better management. So what are we going to do next? We're going to focus on a little bit more structural genomics, and typically we're going to keep on going with this identification of the source allele corresponding herbicide resistance. So I don't believe per se that resistant alleles come specifically from Lolium rigidum, Lolium perenne. But what I believe instead is that we have occasionally, when you form a hybrid, you've got some level of aneuploidy, and this aneuploidy promotes more breaks than not, and these breaks are being filled by whatever the plant finds, and in that case, copy number variation. And if the copy number increases in favor of herbicide resistance genes, then it's particularly beneficial for plants. And this is really a model I'd like to further investigate. At the population level, we're going to continue our effort working at different scales in terms of landscape scale to define the best management units, because still unclear at the Australian level at which scale we need to manage resistance. Certainly not at the single paddock level based on our data, but what is the right scale is still an open question. And we set in place a mesocosm experiment with a big 130-liter tub filled in agricultural soil that we repleted with all sorts of genetic variation from ryegrass population collected over the landscape. And we've planted wheat, and we're measuring response to selection in response to different management treatment. We're trying to have a bit of a more agronomic approach where we combine herbicide, different row spacing, different level of soil perturbation to see how over time across multiple growing season, how all ryegrass populations are being selected for. So this was made possible thanks to my lab and the awesome work mostly driven by Jeff Peril, who's managing the project. Now has moved on since last year to take on his own position, where he's now more working on pasture crop and forage crop breeding, so has abandoned a little bit the wheat field. But CJ has since taken over the development of bioinformatics technique, and has been incredible in the last 3 months in generating new results and pushing the reflection. I'm also thanking all the people that have provided at some point some technical help from my lab. Ben, Tanya, Tara, Patty, Emma, and Dika have been really pivotal in generating the data and maintaining that. So most of our program is funded by the Australian government through the Department of Agriculture, Water and the Environment. And we also receive sizable support from the Grain Research and Development Corporation, which is our main industry partner in Australia. Well, thank you for paying attention to my talk, and hopefully if you've got any questions, feel free to shoot me an email. I'd be delighted to engage on the topic. Thank you very much.